Hello friends, today I'm going to cover a new topic and the topic is the bioremediation of plastic by microorganisms, an update. I will cover the following contents in my topic. We can't imagine our life without plastic. Each and everywhere we are using in different situations, different type of the plastics in different packaging material like that. Why the plastic is used? Because it has a wide variety of plasticity, and it is basically non-degradable, persistent, and the pollution is caused just because of recalcitrant in nature. The history of plastic, the major history is by Edward Seaman in 1983. This is a German apothecary discovered polystyrene, and then in 1909 the discovery of bakelite was there, and then in 1935 the discovery of polyethylene, low density polyethylene was there. So it was a very accidental discovery. The scientist uh, Reginald Gibson and Eric Fawcett were experimented on the ethylene, and high pressure experiment on the ethylene, and what he observed in next morning, that due to the leakage of the vessel, the ethylene was reacted with the oxygen, and finally he was getting uh, waxy material, and this waxy material, this transparent material, was polyethylene. So this is all about the chronology of the plastics development, plastic invention, different type of plastic production. So in 2015, it was about 300 million metric ton was plastic produced. In gradually, it will increases. In 2020, it was 360 million metric ton, and the India was producing about 3.46 million tons of plastic in 2020, and if the production is still going on, definitely in 2020, in 2050 it will become 1000 million metric ton. During 1950 to 2022, the total production, out of total production, only 21% is still in the use, 9% is recycled, 12% burnt, but 58% is used as landfills, and it pollute the earth as well as the marine. So different type of the plastic and its uses are the polyethylene terephthalate, polypropylene, high density polyethylene, low density polyethylene, polyvinyl chloride, and polystyrene, and according to their different properties, they are having different uses. Due to the complex structure of different plastics, they are having in different use, the polyethylene, they are having 36.3% and highest percent in the use, while the others are lower side, like the polystyrene is very less used, 7.6%. It is just the complexity in the structure, because the polypropylene, polyethylene, polyvinyl chloride, they have carbon-carbon bondings, while the polyethylene terephthalate and polyurethane having the carbon-oxygen bonding. So due to these bondings, they are called as thermosets, and carbon-carbon bondings, they are called as a thermoplastic, because they have plasticity, and uh, due to the plasticity, they are having higher use, like 76%, and due to the lack of plasticity, only 18.4% is used. They are having different ecological disturbances, like 6.3 billion tons of plastic waste is there, and about 72% is used in the landfill, while 14% is used in the incineration. Due to the incineration, it leads to the global warming, human toxicity, and acidification of soil. And due to landfilling, it will leads to pollution of different ecosystem like marine ecosystem and soil ecosystem, and it decomposes into the landfills. And the plastic in higher in size, causing different physiological effect in the soil as well as in the human and animals, while uh, there is decreasing in the size leads to chemical effects in the animals and ecosystem like toxicity and growth reduction in the invertebrates, etc. So, in the animals, there is one postmortem I conducted in the field, and I got the death of cow cough is just due to ingestion of plastic, and I found the plastic into the rumen. Day by day, the plastic we are ingesting and inhaling, according to the United Nations Environmental Program, 75 particles per day we are taking inside outdoor, while 3000 particles per day inhaled and ingested at indoor. And in 250 ml of water, 49 particles we are taking. In 250 ml of beer, 27 particle we are taking. In 20 gram of sugar, 1 particle is taking. And 20 gram of honey we are taking 30 particles of microplastic. And 20 gram of salt we are taking 14 plastics, microplastics. The plastic also acts as a inner material, so that it also carries different bacteria and viruses. Like recently we got COVID-19 virus in a one research article, and Vibrio species that is also carried by this microplastic. So, according to the United Nations Environmental Program, there are two methods of taking the microplastic: is ingestion, and inhalation leads to the presence of plastic particle found in the whole body. And recently, the human placenta also contains microplastic, and these microplastic leads to different disturbance in the physiology of the organs like neurodevelopmental disorders, hormonal disorders, respiratory disease, cardiovascular disease, metabolic disease. Then there is decrease in reproduction performance, and there is a decrease in the birth weight, and there is delayed in puberty, etc. In different experiment, the in the mice is also getting infected, and due to the plastic, and it may leads to heart failure, muscle damage, and the reduced production of the blood from bones. There is abnormality in the brain, embryo development is reduced. There is different polyovarian syndromes. There is a decrease in the spermatogenesis, and it is found in the gut, so leads to decrease in the absorption, and damage in the gut microflora, damage in the gut epithelial cell, reduce in the liver size, etc. So the same things is seen into the marine ecosystem. Now, the my main topic is the plastic biodegradation. The plastic degradation is of two types, like biotic and abiotic. Abiotically, it is degraded by naturally with UV radiation, heat, and mechanical breakdown, and abiotically by man-made, like through recycling, pyrolysis, incineration, and landfilling. Biotically, it is fragmented by invertebrates and microorganisms. So these are different abiotic factors like sunlight, temperature, water amount, and oxygen level. But It will also affected by different physiochemical properties like molecular weight, crystallinity, structure, hydrophobicity, additives, functionalization group, and production technique. If there is a high in the molecular weight, definitely the low in degradation, high in crystallinity, high in structure complexity, high in hydrophobicity, high in quantity of additives, functionalization group is more compact, and different type of production techniques leads to more complex to degrade. Abiotically, I already described. Now comes to the biotic degradation. So first, the biotic degradation of the plastic is done by invertebrates. They are found in the soil as well as in the marine ecosystem. The invertebrates is having different gut microbes. They are chewing the plastic, and they reduce the size of plastic, and finally they are degraded by they are consumed by gut microbes, and the microplastic and nanoplastics comes out into the feces. Experimentally, these are different invertebrates. They are found marine in terrestrial ecosystem. The duration is about in given duration, this different type of microplastics is degraded from this size to this size, and different family are involved like Tenebrionidae, then Pyralidae, then Bostricidae, then Tinidae, and Circulinidae. These families are involved in the plastic degradation, and they are having the gut microbes like Bacillus, Staphylococcus aureus, then Enterobacter, Pseudomonas, Aspergillus, etc. And these are involved in different plastic degradation like polyethylene, polystyrene, like that. So these invertebrates are only responsible nowadays, and these different bacteria and fungus are found in only invertebrates. Now the research are going on for vertebrates, because the gut microbes of vertebrates is not capable of degrading plastic, and research are going on. So these are different invertebrates, and they are found in different ecosystem like terrestrial, and they are consuming this type of plastic, and these of the following bacteria are present inside of the gut. So out of the 100% invertebrates, 29% terrestrial insects were found to capable to degrade the plastic, and 71% of marine insecta are found to capable degrade the plastic, and these are the ratio of plastic degradation by this insecta. Out of them, 40% of polyethylene is degraded by the insects. So after the chewing mechanically, this plastic is degraded into the microplastic and nanoplastic. Further, there is biotic degradation is done by the microbes under the following four categories, four steps. These are the biodeterioration, biofragmentation, assimilation, and mineralization. Before going to biodeterioration, the bacteria colonize to the plastic material with the help of biofilm or with the help of flagella, and after that they attach to the plastic surface, and it will deteriorate the plastic surface. Then the biofragmentation is the method in which the plastic is converted into the monomer and oligomers. Uh, this all biofragmentation is done outside of the bacteria. Then after conversion of the monomer and dimer, it is assimilated by the bacteria, and inside of the bacteria, endoenzymes is responsible for mineralization into the carbon dioxide, and water is produced during the TCA cycle. And this mineralization is done aerobically and anaerobically. The aerobic bacteria digest into the carbon dioxide, water, and anaerobically it is there is production of methane, carbon dioxide, and energy. And the main responsibility to digest the plastic is just because of production of the enzymes by microbes, and these enzymes are acts on substrate and finally degrade into the monomers. These are mainly the PETase enzymes, esterase enzyme, and cutinase enzymes. And further, these enzymes convert into uh, the monomers, and finally it is mineralized under the TCA cycle, and there is a production of carbon dioxide and water. So in 2019, recently there is one bacteria, that is Ideonella sankiensis. It is uh, discovered by Sakai City by in the Japan uh, in 2016, and this is also known as the plastic-eating bacteria. This bacteria uh, this bacteria produce their enzyme, and with the help of flagella, this enzymes is the PETase enzyme, and this enzymes works on the plastic film. It is responsible for the degradation of this plastic, and this is the electron microscopy photograph of Ideonella sakiensis. So this bacteria attached to the plastic surface with the help of this flagella, and the plastic is before degradation is giving insight, and after degradation is looking in this picture. This bacteria attached to the biofilm to the plastic surface, and it produces PETase and metase enzyme, and further this polyethylene terephthalate degraded into the ethylene glycol and terphthalic acid. So this is whole degradation process by Adenoella sankiensis. Here the PET is degraded by exoenzymes. Finally, this monomers is getting inside of the bacteria, and this is a mineralization procedure. And finally, there is a production of water and carbon dioxide. So this is whole biochemistry of this uh, PET degradation. This is another bacteria called as Rhodococcus viewer C208. This bacteria is responsible for degradation of plastic, especially the polyethylene and polystyrene. And under an experiment, it shows that 8% reduce in the molecular weight or the weight loss within 30 days in the polythene, and 0.8% weight loss seen in polystyrene after 8 weeks of incubation. So this is a bacteria, Rhodococcus ruber, under the scanning electron microscopy. It shows in the A and B there is 7 days growth, and sorry, it is 16 hours growth incubation during the incubation, and after 7 days of incubation, this is a growth seen on the film, that is on the uh, polythene, that is seen under the electron microscopy. This is another bacteria, that is common as acidovirens TB35, and this bacteria produces pu esterase, and it degrades the polyurethane, and it converts uh, polyisocyanate. This bacteria also release urease, esterase, and proteases, and it further degrades these monomers. This monomers is assimilated and taken up by bacteria. Further, it will produce ethylene glycol, and that enters into the TCA cycle, and that will give the energy to this bacteria. There are different type of bacteria, and uh, uh, they are discovered, and these bacteria are responsible for degradation of such type of plastic like polystyrene. They are found in different ecosystem. Now these bacteria are enlisted here. They are producing different type of the enzymes, and these bacteria are responsible for degradation of polyethylene within these given incubations, and there is weight loss is given here. These bacteria enlisted here are responsible for degradation of PET, that is the polyethylene terephthalate. In a given temperature, this enzyme works, and during this incubation period, there is weight loss is seen. The bacteria which is responsible for degradation of polystyrene, they are enlisted here with their given enzymes. They are isolated from different areas like in soil and from gut, and they are having different on the different incubation period leads to degradation of the plastic, and there is weight loss. This bacteria degrading the polyvinyl chloride. This bacteria degrading polypropylene. This bacteria are degrading polyurethane. These are the fungus enlisted here having different enzymes, and different plastics are degraded by this fungus. So this is a list. Now comes to the next organism, that is algae. Recently it is discovered, that is discovered by University of Madras and Presidency College, Chennai. And the name of this algae is Uronema africanum, because it is found in the river uh, in the Asia, Europe, and Africa. That is not river. That is actually it is called as ocean. Huh? That is common in Asia, Europe, and Africa. So it is called as Uronema africanum. So how the degradation is to be measured? The degradation of a plastic is generally measured by change in the functional group due to enzymatic degradation. There is change in the hydrophobicity or hydrophilicity of the plastic material. There is change in the surface topography. Mechanically, there is change in the plastic strength. There is change in the uh, molecular weight of the plastic leads to degradation. The change in the weight is measured by weighing the material of plastic and leads to weight loss. Topographically, the measurement is done by the scanning electron microscopically, and then chemical bonding is measured. The change in the chemical bonding is measured by Fourier transform infrared spectroscopy. Then there is change in the hydrophobicity is detected by bath test. So this is all about the test that is carried out, or the different protocols that is carried out under the laboratory condition. Finally, on the petri dishes, these bacterial colony is seen, and where the plastic material is there, there is a clear zone is seen. That means there is assimilation of this plastic material. If the plastic material in the uh, uh, form of granules, so there is change in the shape of this granule. You can see here this is a whole granule, and after incubation there is change in the granular structure, leads to degradation. So there is how can we measure the uh, plastic degradation by change in the shape, change in the size, change in the molecular weight, change in the hydrophobicity, like that. The molecular weight is measured by this formula. There is loss of weight is measured by initial weight minus final weight upon initial weight. So this is the plastic under the electron microscopy before degradation, and this is the plastic adhered with the biofilm that is produced by the Pseudomonas aeruginosa. Pseudomonas aeruginosa is majorly worked and found that it is a great plastic degrading bacteria. The surface hydrophobicity is measured by bath test, and in this test, different buffer system is used, and the bacteria and the in the test tube the bacteria and hexadecane is mixed to each other, and finally this suspension is bacterial hydrophobicity and plastic hydrophobicity is to be measured by hexadecane. In the test tube, a different mixture of hexadecane and bacteria is mixed, and finally, the measurement of OD of the aqueous suspension with the spectrophotometrically taken. A cell free buffer maintained as a reference as a blank. Then the chemical bonding is measured by Fourier transform infrared spectroscopy. In this method, the chemical bonding is having different angstrom in size. There is a carbon uh, distance, carbon oxygen distance is there. So this distance is getting weaker, and this is before um, treatment of the bacteria, and this is after the incubation of bacteria. So on the same infrared, same zone of infrared showing different strength of the carbon carbon bonds leads to degradation. We can also measure this bacteria by phylogenetically, that is this bacteria responsible for degradation or not. Now comes to the final conclusion that pollution is caused by plastic, and it is just because of non-degradability, persistence, and calcitrance in the nature. Uh, Emerging plastic pollutants in the nature are explicit and linked to the various chronic detriments in the biotic component. Some microorganisms, especially bacteria and fungi of garbage soil, sewage water, soil of dumping site, are efficient to degrade plastic and utilize plastic as a carbon source. Then some enzymes which are responsible, like cutinase, protease, lacase, they are responsible for degradation. They worked in a specific uh, temperature, pH, contact time, and concentration. These enzymes are responsible for biodegradation, and why we are using these enzymes, because they are eco-friendly, safe, and economical. Introduction of bioengineered microorganisms in the today's research enhances the degradation of plastic more effectively and efficiently. Based on this knowledge, genetic engineering approaches to create recombinant microbial strain and or enzymes could be adopted as a preferred strategy to enhance the biodegradation of plastic waste. So, looking all these things, the further future prospect is that uh, because the level of protease enzyme is very low in normally Idiomela senkensis, so there is a need to increase the expression of this protease enzyme for the hydrolysis of PET and there is need. So definitely in the future it is especially the scientists will focus on the PETase enzyme for hydrolysis. Extraction of these enzymes and their modification in order to increase their enzymatic activity has been one of the main research area in order to cope up with ever increasing plastic pollution in the environment. With the advancement in the microbial genetics and molecular biology, the development of genetically tailored microbes become the practical feasibility to degrade the complex polymers in the environment. A cocktail of two or more enzyme enhances biodegradation. The manipulation of active site of enzyme is one of the methods that can be employed to enhance the stability of plastic degrading bacteria so that these enzymes can work in adverse environmental conditions including hot climate and extreme cold climate as well. Site-directed mutagenesis is one of the methods that can be employed to alter the structure and functional group of the active site of the enzyme so that it can accommodate plastic polymer with ease and can functions with wide range of temperature and pH. So these different microbes will be genetically engineered and the genetic engineering with the help of plasmid transformation will work to produce a lot of concentration of different enzymes leads to degradation. And this is a concept of bioreactor in that the plastic is first degraded and it will chewed and it will convert into the microplastic. This microplastic will go inside of the chemical treatment with citric acid and potassium permanganate and HCl, and finally this chemically treated uh, solution will reach inside of this biogas chamber. Here we will put the different bacteria, fungi and algae solution that will work on this plastic, and there will be production of the methane and carbon dioxide, and this will be further captured for the energy purpose and for further biofuel. So this is all about the references. So thanks for watching this video. I just want to say that reduce the use of plastic, because ultimately this plastic uh, will become reduce the lifespan and it will destroy the ecosystem. So that is my humble request to please reduce the plastic use and use the biodegradable plastic only. Thanks for watching.